attention, and this will help you for your test tomorrow. So let's take a look at what we have here and open up behind my document here. I do have the keynote, so that should help you a great deal in determining what we're talking about here. But let's first look at our reactants of photosynthesis. It's carbon dioxide, and please make sure that you know that that's CO2, water, and light. Water, again, is H2O. So that way you understand that it could also be represented as the letters or the chemical formula. It's just much easier to type it out as we go along. We know that our product from photosynthesis is glucose and oxygen. Again, glucose is C6H12O6. So make sure that you have that written out. Also, for the equation, you want to make sure that you have 6 carbon dioxide. Again, that would be written 6 CO2, 6 water. That would be plus 6 water. So we could put the plus sign there. Plus 6 water, which would be 6 H2O, plus, and don't forget the light. In our notes, the light is depicted as a big sun. And this is going to make glucose, which we already talked about above, as C6H12O6. You get one of those. And 6 oxygen, or 6O2. So I want you to have that written in your notes there, both as words and as symbols. And the energy use here is to make glucose. The purpose of the energy that we're using here is to make glucose, to make food. Now, where do these things take place? Well, the light reaction, which we'll talk about in just a minute, takes place in the thylakoid membrane. The Calvin cycle, which is the second stage of photosynthesis, takes place in the stroma, which again is the liquid portion. There are 2 stages of photosynthesis. First is the light dependent. Remember, if you're dependent on something, you need it. So light dependent needs light. It uses light to make energy carrier molecules. And we talked about these before. The next step is the Calvin cycle. And the Calvin cycle uses the energy carrier molecules from step 1 to make glucose. This is where our glucose is made. So that's what happens in photosynthesis. Now let's go up here and fill out our cell respiration. Now, cell respiration, our reactants, or what we start off with, is glucose, again, C6H12O6, and oxygen, which is O2. Our products, or what we end up with out of this, is ATP, which we know as our energy molecule, carbon dioxide, which is CO2. Again, I want it written both ways for you. Oh, I'm having trouble typing here. It's losing in that space there, so let's bring it down here and see if we can. Carbon dioxide and water. Again, carbon dioxide is CO2, water is H2O. So now let's take a look at our formula or our equation. We start off with glucose, which is C6H12O6, and then we add 6 oxygen, and it makes 6 carbon dioxide, plus 6 water plus 1 ATP, which is our energy molecule. All right? And so what is the energy used for here? It takes glucose and it makes ATP out of it, which is a smaller quantity of energy so that we can use it more efficiently. So where did the different stages of cellular respiration take place? Well, glycolysis takes place in the cytoplasm of the cell. The Krebs cycle takes place in the matrix of the mitochondria. And the electron transport chain takes place in the mitochondria, which is more of the folds of the mitochondria is where that takes place. So let's talk about the stages here of cellular respiration. We have 3 stages. The first stage is called glycolysis. And glycolysis is anaerobic, so you need to know that, which means it does not need oxygen. It makes a little ATP. All right, so it doesn't make a lot of ATP, but it makes a little bit of ATP without oxygen, anaerobic. And what it basically does is it takes glucose and breaks it down into pyruvic acid. So it's pyruvic acid is what it breaks it down into. All right? And then in the second part, which is called the Krebs cycle, what happens in the Krebs cycle is we take the pyruvic acid and we make electron carrier molecules. Sound familiar? We had it in the other ones. It makes electron carrier molecules. We get a little ATP in this cycle, and it is aerobic, which means it has to have oxygen to it. The third and final stage is electron transport. Electron, let's see if I could spell, that would be good. Transport. And what happens here is that it is aerobic, makes lots of ATP in this section here. And this is where it uses the electron carrier molecules from the Krebs cycle, okay? So it uses those molecules that were just created. Right? So those are the 3 steps. The other thing on this is that we need to know what our ATP is made of. And it is made up of adenine, ribose, which is the sugar, and it is made up of 3 phosphate groups. Thus, we call it triphosphate. Now, this other one, adenosine diphosphate, is made up of adenine. It is made up of ribose, which is sugar. And it is made up of 2 phosphate groups. Okay, versus the 3. Everything on this comparison sheet is really important for you to know, and you're going to need to know it for the test. So with that said, what I really want to do is take a look at the notes here for a second, because we've talked about glycolysis being anaerobic and not needing oxygen. So let's take a look and see what happens when we don't have oxygen present. So if oxygen is not present, remember that very first part of photosynthesis, or cellular respiration, we break down without using oxygen. But after that, to be able to produce our ATP, we need to have oxygen. There are two types of anaerobic respiration, two ways that we can make ATP without oxygen. You don't get a lot of it, but it's ways that we can use it. First is called lactic acid fermentation, and this produces lactic acid, and that's why your muscles get sore. If you overdo it and you have what we call an oxygen deficit, we still need the ATP to keep going, and so it's made by undergoing fermentation. And by undergoing fermentation, we do produce some ATP to keep us moving, but the byproduct of that is lactic acid, and it will build up in your muscles, and it will make your muscles very sore. The second type is alcoholic fermentation, and this produces carbon dioxide, alcohol, and ATP. It is performed by yeast and some other bacteria. And so this is how we make a lot of food products, by using this form of fermentation. So what happens is you have your pyruvate and you have no oxygen, so you're either going to have to use your alcohol or ethanol or lactate. If you do have oxygen, it's going to go in and it's going to start with the Krebs cycle, and then it's going to go on to the electron transport chain. So that way you have an idea of what's going to happen whether you have oxygen present or not present. Also, please make sure that you know all of your prefixes for your quiz tomorrow. The first prefix is chromo, which means color or colored. Goes along with what we're talking about pigments in our plants, etc. Phyll, P-H-Y-L-L, which means leaf. Again, goes along with what we've been talking about with photosynthesis. Tel or tele means distant or end. Sperma or spermato means seed. And then lastly, phyte, P-H-Y-T-E, means plant. So all of these are prefixes that you want to take a look at tomorrow and that you will need to know for the test. I hope all this helps you. Happy studying.